Assalamu alaikum viewers. I, Dr. Sadaf, welcome you all on this platform of online university courses OUC. This platform is actually designed to help university students in understanding difficult topics, especially conceptual ones. So I am here to help you in different biology related and helping you in different biology related topics. So today the topic which I have sought for you is urinary system. So let's begin with it. Urinary system, as you all know, is its other name is renal system or urinary tract. The point to be noted here is very important, which you all already know, that urinary system is one of the organ system, and these organ systems are developed in higher organisms. That is, more specifically, they are developed in good state in animals. Though present somehow, some of the organ systems are present in plants, but they are not that much developed. For example, plants though respire, but they lack a proper breathing system, unlike animals. Similarly, they have a transport system in the form of xylem and phloem, but plants lack cardiovascular system. Similarly, they lack nervous system, and so on and so forth. So urinary system is only a property of higher level organisms, that is animals. So let's see, urinary system is comprised of which organs. Renal system or urinary tract, it consists of the kidneys, a pair of kidneys as you can see on the slide, a pair of ureters, bladder and a urethra. So pair of kidneys, pair of ureters, and single structures are bladder and urethra. Excretory system, as you see on the slide, it is a biological system that removes excess unnecessary materials from the body fluids of an organism. You know, excretion is a process in which unnecessary materials are removed from the body of an organism, and it is a part of homeostasis. What is homeostasis? To maintain a constant internal environment despite of changes going on in the external environment. So homeostasis is again a property of higher level organisms, and they can maintain homeostasis in many ways. There are three different components, you can say, of homeostasis. One is excretion, like whenever waste products they try to accumulate into the body of animals, they just get rid of it through their excretory system, so that internal body system, it remains at equilibrium. Nothing unnecessary is going to be piled up there. Then another component of homeostasis is thermoregulation, that is to maintain a constant temperature. You know, we as human beings are actually social animals. We can maintain our body temperature at 37 degrees centigrade, or in other words 98.6 degree Fahrenheit. So despite of the fact that we are in very cold environment or we are in very hot environment, although the external temperature may fluctuate, but our body tries to maintain our temperature at a constant level in equilibrium. Similarly, there is another component of homeostasis, that is osmoregulation. In this method, the osmolarity of body fluids of the organism is actually maintained at a constant level. It is at a level which is usually decided by the nervous system of that organism, and every organism has a different. Some are hypertonic, some are hypotonic, some are isotonic to their environment, so on and so forth. So urinary system is actually excretory system, and it actually tries to maintain homeostasis in organisms by removing excess unnecessary materials from the body fluid of an organism, and thus maintaining a constant internal environment to prevent damage to the body. Now, what are kidneys? Kidneys are actually a pair of excretory organs situated on the posterior abdominal wall, one on each side of the vertebral column behind the peritoneum. So what is peritoneum? All the organism in the all the organs and organ systems which are present in the abdominal cavity, they are actually suspended and covered by a double layer membrane which is known as peritoneum. Like, small intestine and large intestine are literally hanging in the abdominal cavity, and they are just covered by peritoneum. In this way, they are free to move. But unlike these, kidneys, a pair of kidneys are just present against the posterior abdominal wall. It means their posterior or back surface is just adherent to the posterior abdominal wall, and anteriorly they are covered, or on the front they are covered by the peritoneum. They're present on each side of the vertebral column, and if you noticed here in this diagram, the right kidney is slightly lower than the left kidney. The reason being, there is liver on the right side, so it pushes the right kidney slightly lower into the abdominal cavity as compared to left kidney, as there is no liver on the left side, so left kidney is slightly higher than the right one. The two adrenal glands, you can see there are two adrenal glands which are present above the kidneys. Okay, so now if we cut a kidney into a longitudinal section from above to below, so we can see three different distinct regions in kidney. The outermost region is known as cortex. In the middle there is medulla, and then in the center, still inner to the medulla, there is a renal pelvis. It is in a region which is known as hilum of the kidney. So as kidneys are bean-shaped structures, you have noticed, so just like bean, they have one concave and one convex surface. The convex surface is actually facing outside, and the concave surfaces of both kidneys are facing towards the vertebral column. And this concave surface is also known as hilus or hilum. As you can see here, the two kidneys, they are lying on either side of the vertebral column, facing towards their concave surfaces are facing each other and also facing vertebral column, while convex surfaces are facing outside. In the cut section, as you can see, the outer portion is known as renal cortex. Brackets are indicating its thickness. It is much less in thickness as compared to the inner broad part, which is renal medulla, and still inner the lighter color pelvis, which is just emerging from the hilum or hilus. As you can see here, hilum or hilus, not only from the hilum, pelvis, it comes out, and later on pelvis continues down as ureter. But at the same time, blood vessels, you can see arteries and veins, renal artery and vein actually, it is entering into the kidney through this concave surface, that is hilum. At the same time, a renal nerve also enters into the kidney from here. So renal cortex, renal medulla, and then the pelvis. Now if we discuss one by one, so we see after kidneys in the small diagram, you can see from kidneys are emerging ureter. Ureter is a tubular organ about 25 centimeter long, and it begins as a funnel-shaped renal pelvis. Above is renal pelvis, and it is funnel-shaped because just like the funnel, it is broad at the upper end, and as it narrows down as it continues into ureter. So each ureter extends downwards parallel to the vertebral column to join ultimately the urinary bladder. And what is urinary bladder? Urinary bladder is a hollow distensible muscular organ. There is one bladder, as you can see here. There is only one bladder, although two kidneys, two ureter. Ultimately, they bring urine after formation into the bladder, and bladder is a hollow distensible muscular organ. Whenever it is filled with the urine, its muscles relax simply. But urine, as it exerts pressure on the bladder, ultimately during voiding urine or micturition, during the process of micturition, its muscles contract. So it is simply a storage place for urine, and it acts as a urine reservoir. Then there is a single tube urethra that carries urine from the urinary bladder to the outside of the body. Let's study in detail the structure and function of kidney. Actually, the branch of science or a branch of biology which deals with structure, function and diseases of kidneys is known as nephrology. So anything discussing something about kidney, it comes under the heading of nephrology. Each kidney, if we cut it, we can see it is a reddish brown bean-shaped organ with a small surface area. A fibrous connective tissue called a fibrous capsule also encloses each kidney. Lateral surfaces, as I told you, are convex. Medial side is deeply concave, and the resulting medialmost depression actually is it leads into a hollow chamber, also known as renal sinus. And entrance to this sinus is termed as hilum. So this hollow space is sinus, and its entrance is known as hilum. And hilum is the place where renal artery, renal vein and nerve, renal artery and renal nerve actually enters, and renal vein and ureter it exits from the kidney from here. Now if you see in medulla there are many cone shaped or pyramid shaped structures. You can see these are also known as renal pyramids. The base of each pyramid is actually located at the boundary between renal cortex and renal medulla, and the tips of the pyramids are actually known as, their tips are facing towards the hilus, and tips or narrow points are known as renal papilla. Now step by step, as you can see in this diagram, from the renal emerging from the renal papilla are small passageways or tubes which are known as minor calyx. Plural is calyxes. We say many minor calyxes ultimately join to form large tubes known as major calyxes, and major calyxes ultimately empty into renal pelvis, which is the upper end of ureter. So inside each renal pyramid, the structural and functional unit of kidney, that is nephron, it is present. So many nephrons are present. About 1 million nephrons are present in one kidney. So they are distributed within these 10 to 12 pyramid-like structures. Now within each pyramid there are many nephrons. Ultimately urine from the nephrons, it collects, it comes out through the papilla, through a papillary duct into the minor calyces, then from here into the major calyces, from here there to into renal pelvis, and ultimately through the ureter into the bladder and stores there until micturition. So now what is nephron? Here you can see the comparison. On the left side there is a picture of cut section of a real kidney of sheep or goat, and right side it is a diagram. So as we can compare, it is not as clear, every structure is not as clear as it is seen in the diagram, but still we can appreciate outer region which is cortex, inner pyramids can also be seen, then minor calyces emerging from the papillary region of each pyramid, then they join to form major pelvis, then a pelvis, and ultimately ureter. Okay, so now what are nephrons? A nephron is a structure, is the structural and functional unit of kidney, and each nephron, the point to be noted here is that each nephron is not a single cell, rather it is a structural functional unit of kidney, but each nephron is made up of many small cells which are usually cuboidal epithelium. It lines each nephron. Each nephron we can divide, they take actually blood, metabolize nutrients, help pass out waste products from filter blood. And as I have already mentioned, each kidney has about 1 million nephrons, and each has its own internal set of structures. Now broadly we can categorize each nephron into two main parts. A non-tubular part which is not tube-like, this is known as renal corpuscle, and later on its tubular part which is tube-like structure is known as renal tubule, as its name indicates. We can further divide its first non-tubular part, that is renal corpuscle, into two further regions, a network of capillaries or a bunch of capillaries, that is glomerulus, and a cup-like structure just enclosing these capillaries known as Bowman's capsule. Tubular part again has three regions, 1, 2, 3. Number 1 is proximal convoluted tubule. Proximal means which comes first. So proximal convoluted, and convoluted means having convolutions or folds, zigzag. So proximal convoluted tubule is that part of tubule which comes soon after the Bowman's capsule. Then there is a loop of Henle, as its name indicates, is in the form of a loop. It has a descending limb and an ascending limb. Descending going down, ascending means going up. And then comes distal convoluted tubule. Distal means that which is at the distant region, far away. So distal convoluted tubule is at the end. Again, it has many convolutions or folds. Now you can see appreciate all the structures in this diagram, that each pyramid, on the left side you can see each pyramid and only 2 nephrons are mentioned here, although there are many nephrons within each pyramid. As you can see, first part is renal corpuscle. As I mentioned, a corpuscle is again divided 2 different parts, red colored glomerulus or glomerular capillaries, and which are enclosed by a cup-shaped structure, that is Bowman's capsule. Both of these, glomerulus plus Bowman's capsule, both of these make renal corpuscle, that is first part of glomerulus. Then comes the renal tubule. As you can see, proximal convoluted tubule, it's labeled here. Then comes loop of Henle, a descending limb and ascending limb, and then distal convoluted tubule, and ultimately distal convoluted tubules of many nephrons are emptied into one duct, which is known as collecting duct. So one collecting duct, it, as you can see also on the left side, that here 2 nephrons are being shown. They are emptying into one duct, that is collecting duct. It is also showing many cut ends, means many other nephrons are going to join it. So many nephrons ultimately empty their components, that is urine, into one collecting ducts. Many collecting ducts join and they emerge from the narrow end of each pyramid, that is papilla, and they ultimately empty into one papillary duct. Papillary ducts, I told you earlier, empty their components into minor calyces, then comes major calyces, then pelvis, and ultimately ureter. So as you can also see here, a loop of Henle, it has some thick part and a thin part. Thin part, it has its own function, thick part has its own functions. Now comes blood supply, blood circulation to nephron. As you can see, an artery which is supplying the kidney, it is known as renal artery, and actually it is a branch of descending aorta. So renal artery, ultimately after entering into the kidney, it divides into many interlobar arteries and interlobular arteries. Each artery actually encloses pyramids and it gives branches around or along the pyramid. So ultimately it divides into smaller and smaller branches, which are known as arterioles. Ultimately arterioles, they divide to form glomerulus, which is a bunch of capillaries, and ultimately from the glomerulus arises another arteriole, efferent arteriole. So there is a twist here, because renal arterioles, they are different from rest of the body arterioles. As you can recall, you must have studied in early classes that each artery in the body, there is a stepwise process. Each artery is a large structure. First of all is the aorta, which is the largest blood vessel in the body. It divides into smaller arteries. Arteries divide into smaller branches, which are known as arterioles. Arterioles ultimately divide to form capillaries, and many capillaries, now the back process starts. Capillaries are the smallest blood vessels which provide oxygenated blood to different tissues and organs. Many capillaries ultimately join to form venules, and venules actually carry deoxygenated blood. Many venules join to form veins, and ultimately veins join to form, empty their components into inferior or superior vena cavas, and vena cavas ultimately enter into the heart. They carry the deoxygenated blood back to the heart. But here the case is different. Renal artery, it divides into interlobular branches. They divide into arterioles. Arterioles once divides into glomerulus, which is a bunch of capillaries. Ideally, after capillaries have been formed, they must join to form venules. But here, glomerular capillaries, they are again joined to form another arteriole, as you can see in the diagram, because arterioles carry oxygenated blood, which is being shown as red. So again, blood vessels emerging from glomerulus are not blue. They are not veins or venules. These are again making efferent arteriole. This efferent arteriole, again, it surrounds different parts of nephrons, tubules, and they form another bunch of capillaries known as peritubular capillaries, and these capillaries join ultimately then to form different veins or venules, and they join just like parallel to the interlobular arteries or interlobular veins, and then they join to form a renal vein. A renal vein then emerges from the kidney, taking the deoxygenated blood, emerges from the hilum. Why this happens in kidney? There is a logic behind it, because every time capillaries actually are very narrow blood vessels, and as their area decreases, pressure increases in capillaries. Usually capillaries are so narrow that blood cells have to line up. They have to pass one by one, very slowly. As they pass slowly, oxygen, nutrients, everything, it gets the chance to move from the blood into the surrounding tissues and to supply them. So ultimately, and meanwhile, the tissues which have produced different waste materials and carbon dioxide as a process, as a result of respiration, it moves from the tissues into the capillary blood, and ultimately when they are making at the end region of the capillaries, when they are starting to start joining to make venules. So that's why the color of the blood changes into blue, because of the presence of carbon dioxide. But here is the case is slightly different, because glomerulus capillaries are not designed to supply nutrients to the nephron. Actually, glomerular capillaries are designed to build a high pressure, because the afferent arteriole, afferent means incoming, that's starting from afferent means incoming and efferent means outgoing. So afferent arterioles, which brings oxygenated blood into the glomerulus, and it is gradually from the glomerulus till reaching the efferent arterioles, its diameter of the blood vessels or capillaries, it decreases and decreases. As the diameter decreases or area decreases, ultimately pressure increases, because area and pressure are inversely proportional. So where in the presence of very high pressure, what happens, all the components from the blood, they are just filtered out and they start collecting into the Bowman's capsule. So glomerulus has been designed by God for which purpose? Just to filter the components, because the function of the kidney is to filter. So this function is basically carried out due to the presence of glomerulus and its decreasing diameter. Now all the components are filtered here. They did not get a chance to supply nutrients and oxygen to different parts of nephron. That's why these capillaries again join to form another arteriole, that is efferent arteriole, and then this efferent arteriole, it forms a rich network of capillaries known as peritubular capillaries, and through these capillaries blood, nutrients and oxygen, it is being exchanged and being supplied to different parts of the nephron. Now vasa recta, here is the name, as you can see on this diagram, it has been labeled, capillaries are labeled vasa recta also. Vasa recta is a special name given to the peritubular capillaries of some of the medullary or juxtamedullary nephrons. We are going to study it later. These nephrons have very long loop of Henle's which dip deep down into the medulla of kidney, and that's why the peritubular capillaries which accompany these long loop of Henle, those capillaries have been given a special name, that is vasa recta. So I think that supply is clear, must be clear to all of you. Renal artery, it divides a branch of descending aorta, it supplies blood to the kidney. It divides first of all into interlobar or interlobular arteries. Those divide to form arterioles, and then from there arises afferent arterioles, arises glomerulus, then efferent arteriole, then peritubular network of capillaries. Some of these are called vasa recta, not all, and ultimately they join to form venules, interlobular veins, and ultimately in the form of renal vein, and renal vein drains into inferior vena cava, which usually accompanies the aorta and present on the in the abdomen on each side of the vertebral column. Okay, now comes the function of kidney. Okay, function of kidney has actually two different functions. One is, you all know, as we are studying excretory system, so function of kidney is to do excretion, to remove waste materials from the body, and this function is being carried out with the help of urine formation. Second important function is osmoregulation. Osmoregulation, as I told you, is another part of homeostasis, and kidney also plays a very important role in maintaining the osmolarity of body fluids. Body fluids includes blood, tissue fluid, lymphatic fluid, everything. So osmolarity of body fluids is being maintained with the help of kidney. How is it being maintained? Because kidney actually, it is concerned, it controls the formation of dilute urine during the state of hypotonic body fluids, and it forms concentrated urine during the state of dehydration. So how kidney does this function of osmoregulation? This is actually tricky. It is with the help of a hormone which is known as ADH, or antidiuretic hormone. Now how this happens, antidiuretic hormone, if you recall, actually it was being secreted by posterior pituitary. You all must have studied in the endocrine system. So posterior pituitary, actually secretes two different hormones: antidiuretic hormone or ADH, its another name is vasopressin, and second is oxytocin. Both of these hormones are actually produced by hypothalamus, which controls the secretion of pituitary. So what happens, how this function of osmoregulation is being controlled by kidney? What happens when the blood supply to the kidney is it shows kidney that blood is very concentrated, it contains more solute, or it is hypertonic. It can happen in the state of dehydration when a person is not drinking enough water or he is sweating in hot humid weather. What happens, the blood supply to the kidney, actually the blood which is being supplied to the kidney is hypertonic, its osmolarity is more. So this is actually a stimulant, because it the same blood, it when it supplies the hypothalamus, it is a signal, it stimulates hypothalamus to secrete antidiuretic hormone, and hypothalamus, through its neurons and axons of its neurons, it synthesizes that hormone and stores it into posterior pituitary. Posterior pituitary secretes ADH or vasopressin. Immediately vasopressin does two different functions. Number one is it, as its name indicates, vasopressin, it presses blood vessels, or it narrows blood vessels, increasing blood pressure. So when blood pressure is being increased, some other functions are being carried out, they are not concerned with kidney. At the same time, antidiuretic hormones means, diuresis means doubling the amount of urine. Antidiuresis means it inhibits the formation of excess urine. So this hormone actually is going to act on nephron, where this we are going to discuss later. So ADH, when acts on the kidney, it ensures reabsorption of more and more water from the nephrons into the blood. So when more water is being reabsorbed from the nephrons into the blood, what happens, water content in the blood increases. Water is a solvent, and solute part ultimately decreases. So hypertonic blood, it actually, its osmolarity is being brought back to the normal, that is isotonic. It is being set according to the body's level. So in this way, kidney is also performing the function of osmoregulation with the help of ADH. Now we discuss how is urine being formed. Urine formation actually takes place in three different steps. Number one is glomerular filtration, number two is tubular secretion, and number three is tubular reabsorption, and number three is tubular secretion. All of these three steps, they are taking place side by side. It doesn't mean that first of all only filtration is taking place and no tubular reabsorption. No, all of these steps are simultaneously taking place, but we, for our convenience, we have divided into three steps, and we are going to discuss these one by one. Number one is glomerular filtration. Okay, so now as we have already discussed that due to high pressure which is being built up in glomerulus, and why this pressure is being built up, because afferent arterioles, they are larger in diameter as compared to the efferent. The diameter of efferent arterioles is half as compared to the afferent. So gradually the diameter of blood vessels is decreasing as they are moving ahead, and as a result pressure ultimately is increasing. This high blood pressure, it forces all the components from the glomerulus to be pushed and filtered into the next part, that is cup-shaped structure, Bowman's capsule, and this filtrate collects here. Very important thing is that small molecules like water, some small solute molecules, they are filtered out of the glomerular capillaries, including glucose and all the salts, sodium chloride, etc. But larger molecules like proteins as well as red blood cells and platelets, they are not being filtered out. It is very important to note here that in a normal healthy person, there are usually 1 to 2 red blood cells which are present, or almost no proteins or no platelets or no white blood cells are present in normal healthy person in their urine. But if there is a disease, like renal stones or some infections, then red blood cells, white blood cells and proteins might be noticed. So normally they are not being filtered because these molecules are very large in structure, they cannot come out through the holes between the capillaries of the glomerulus. So this filter when it collects into the Bowman's capsule, this is known as glomerular filtrate. So this was the first step in urine formation, that is glomerular filtration. Now once this filtrate is collected into the Bowman's capsule, what happens next to this filtrate? Next is very important step, and that is selective reabsorption or tubular reabsorption. This is a process by which certain substances that have been filtered out of the blood during ultrafiltration, they are reabsorbed. And this is very important step because almost 99% of filtrate is going to be reabsorbed back into the blood. So if this function of kidney, if it would not present, then very large amount of urine, number one, would be produced, and number two, it would be very harmful for the body because body fluids are going to be wasted in this case. So what happens now? This selective reabsorption, it takes place in different regions. Different substances are going to be reabsorbed back into the blood in different regions of the nephron. First of all, we discuss proximal convoluted tubule. And what happens? Actually, almost all the glucose, amino acid, vitamins, inorganic salts, and some of the water, almost everything, it is reabsorbed in this step of selective reabsorption. And how? Because in the proximal convoluted tubule, almost all the glucose and few of the ions, sodium, chloride, they are first of all reabsorbed into the blood. How? Because proximal convoluted tubule, as we have discussed earlier, efferent arterioles just coming out of the glomerulus, they form a network of peritubular capillaries which surround different parts of nephrons, including proximal convoluted tubule. So as the substances, they first of all reabsorb, they have to cross first of all the lining epithelium of the proximal convoluted tubule, which is the cuboidal epithelium. Then they enter into an interstitium tissue, and then comes the peritubular capillaries. And then once they cross a single layer of simple squamous epithelium lining those peritubular capillaries, they enter directly into the blood. So initially, those ions, that is glucose, sodium, chloride, potassium, all of these, they are reabsorbed into the blood, and then a concentration gradient or osmotic pressure is actually built up, which also draws water along with it. As you know, whenever solutes, they come out, they actually, ability of a solute to draw water towards it is known as this osmotic ability. So due to osmotic ability, they also reabsorb or water from the nephron. Water follows these ions ultimately. So from proximal tubule, convoluted tubule, many things are going to be reabsorbed from the distal convoluted tubule. Before that, we have loop of Henle. Loop of Henle, as you can see, it has one descending and one ascending limb. Descending limb of loop of Henle is actually permeable only to water, not to solutes. Because why is it permeable to water? Because of the presence of some specific proteins present in its epithelium, which are known as aquaporins. Aquaporins are actually water related proteins. They facilitate the movement of water from the nephrons from the descending limb of loop of Henle into the peritubular capillaries. So from descending limb of loop of Henle, only water reabsorbs. From the ascending limb of loop of Henle, only salts are reabsorbed, because it is again, now it is impermeable to water. So from the descending limb of loop of Henle, only water is being reabsorbed. From the ascending limb, only solutes are reabsorbed. Then comes the distal convoluted tubule. From here, mixed again ions and water, again it is being reabsorbed into the blood. And then comes the collecting tubule, again water, mainly it reabsorbs water. Now ADH actually acts on descending limb of loop of Henle and from the collecting ducts. So from both of these regions, the reabsorption of water is under the action of ADH, antidiuretic hormone. And solutes which are being absorbed from ascending limb of loop of Henle, those are under the action of another hormone, aldosterone. This aldosterone is actually being secreted by adrenal glands. I have discussed in the beginning, because on kidneys are bean-shaped structures, and there were two glands I have shown you present above the kidneys. Those were known as suprarenal or adrenal glands, and each adrenal gland has two portions, just like kidney: an outer portion, cortex, and an inner portion, medulla. Outer portion, cortex, secretes corticosteroids. Just familiar with its name, cortex, corticosteroids. Corticosteroids are of two types: glucocorticoids, mineralocorticoids. Just we are concerned with one hormone, mineralocorticoids, that is, as its name indicates, aldosterone is one of the mineralocorticoid, and it helps in the reabsorption of minerals, that is sodium, chloride, and many other minerals from the ascending limb of loop of Henle. So this reabsorption actually is under the action of aldosterone, and this aldosterone, its secretion is controlled by suprarenal or adrenal gland. Now, now the same thing is reinforced here. Renal corpuscle generates the glomerular filtrate composed by water, ions, and small molecules. Proximal convoluted tubule reabsorbs sodium, chloride, potassium, water, glucose, amino acids, bicarbonates, calcium, and phosphate, and secretes. Now, the third step, as I have already mentioned, the third step in urine formation is tubular secretion. Tubular secretion is actually a process by which certain substances like ammonium, hydrogen ions, urea, uric acid, all of these are secreted mainly by the tubular epithelium into the loop of Henle. And this process is actually with the help of active transport. Now, as you recall, there are two different types of transport: passive transport and active transport. Passive transport is a transport which occurs according to the nature, that is, from an area of higher concentration to an area of lower concentration. And whenever anything it goes along the nature from higher to lower, it does not need any energy. As we are going along the current, we don't need to put any energy. So passive transport doesn't need any energy, but active transport is opposite of passive transport. It actually occurs against the concentration gradient, that is, from lower concentration to higher concentration. Now, what happens in body? There are many excretory products which are being produced in although in minute amounts as a result of protein metabolism. Ammonia, urea, uric acid, these are being produced. Some of the hydrogen ions extra, they are being produced, although they are in many little amounts in blood. But even keeping that lower amount in blood could be dangerous. So blood has to get rid of those ions. Although through the process of glomerular filtration, many of these ions have already been filtered. They are already present in the nephron, but small amount which is not being filtered, it is left in the blood. Body and blood does not want to keep even that small amount. So body already against the concentration gradient, they are present although in sufficient amount in urine, and as compared to blood has lower amount of those ions, but still body and blood, it with the help of active transport, taking energy from ATP, it pushes those ions and secretes them into the tubular lumen of the nephron so that they can be excreted with the urine. So actually in this the main purpose of this process is to maintain the pH of the urine, also to maintain the pH of the blood, because the normally pH of urine, it ranges from 4.8 to 7.5. So as 7 is a neutral pH, so 4.8 to 7.5 means pH of urine is most of the time it is on acidic side, and this is due to the presence of H ions, hydrogen ions. Acidic pH on one hand, it has two benefits. Number one, it does not allow, or it discourages the growth of bacteria in urine. At the same time, what does it do? As it removes the excessive hydrogen ions from the blood, so it also helps to maintain the pH of the blood. It prevents the blood from getting acidic. So proximal convoluted tubule, from the proximal convoluted tubule, ammonium and creatinine ions are being secreted. Now, loop of Henle, descending limb of loop of Henle reabsorbs only water, and ascending limb of loop of Henle reabsorbs only sodium and chloride. Then thick ascending limb of loop of Henle, as you can see, there are thick and thin segments. Descending limb only has thin segment, while ascending has two parts: a small thin limb and a large thick part. So thick part also, it reabsorbs ammonium, sodium and chloride. Distal convoluted tubule reabsorbs again sodium and chloride plus water, and collecting duct reabsorbs sodium, chloride and water, and also secretes again ammonium and hydrogen ions plus potassium ions. Now there are actually two different type of nephrons. If you see, there are two different type of nephron depending on their location. So the mechanism of urine concentration actually takes place with the help of some particular nephrons, which are juxtamedullary nephrons. And through this process of urine concentration, kidney also controls osmoregulation. How this happens? Let's have a look. As you can see, there are two different types of nephrons: cortical nephrons and juxtamedullary nephrons. Cortical nephrons, as you see, as the name indicates, they are present in the outermost region or cortex of the kidney, and they have, even you can observe here, they have small loop of Henle. As compared to these, juxtamedullary nephrons, actually they start a little lower than the cortical, they start at the junction of cortex and medulla of the kidney, but once they start, they have long loop of Henle's which dip deep down into the medulla, and accompanying these long loop of Henle, the peritubular capillaries are specific peritubular capillaries which are given the name of vasa recta. Other differences we can see in this table. Cortical nephrons are actually, their glomerulus is in the upper region of cortex, and actually 85% of nephrons present in each kidney are of this type, that is cortical nephrons. Juxtamedullary nephrons, their glomerulus is present, as I mentioned earlier, at the junction of cortex and medulla, and only 15% of kidney nephrons, they belong to this category. Their cortical nephrons, their glomerulus is small in size. Juxtamedullary nephrons, their glomerulus is large in size. Loop of Henle of cortical nephrons is small and it extends up to the outer layer of medulla, as in contrast to it, loop of Henle of juxtamedullary nephrons is long, dips deep down into the medulla. Cortical nephrons have ascending limb, thick segment, while most of their ascending limb is comprised of thick segment, while juxtamedullary nephrons have long, even ascending thin segment. Efferent arterioles and they are of large diameter and they form peritubular capillaries, while the efferent arterioles of juxtamedullary nephrons are of small diameter and they are known as vasa recta. The rate of filtration in cortical nephrons is slow, while that in juxtamedullary nephrons the rate of filtration is high. Major function of cortical nephrons is just excretion of waste products in urine, while concentration of urine is the main function which is carried out by juxtamedullary nephrons. Another very important term which is used usually in your books is counter current multiplier mechanism, because this is the mechanism by which juxtamedullary nephrons, they concentrate urine. How does this happen? As you can see in this diagram, descending these juxtamedullary nephrons, they have long loop of Henle, especially descending limb of loop of Henle have long thin segment. Similarly, ascending limb of loop of Henle have long thin segment. So what happens? As you recall, descending limb of loop of Henle, it can, with the help of aquaporins, it can allow reabsorption of water only, while the ascending limb allows reabsorption of solutes only. So due to long descending limb of loop of Henle, a lot of water through these juxtamedullary nephrons, it is being reabsorbed from the nephrons into the vasa recta, or into the blood of the capillaries. When this large amount of water is being reabsorbed, what happens? Although the vasa recta, they are going down, their current is low, their blood, because they are going initially they are going down. But as they go down, more and more water is being added from the nephron into the vasa recta blood. So as this water continues to add up, it increases the volume of blood into the vasa recta. Ultimately, it multiplies the current. Initially, blood flow was slow in the vasa recta, but as it moves down and down due to increased amount of water being reabsorbed from the descending limb of loop of Henle, thin descending limb of loop of Henle, more and more water adds up, more and more volume of blood, so its current multiplies. Although it is going down, its current, ideally, as blood flows further and further away from the heart, usually its volume and its pressure decreases. But in vasa recta, this is not the case, because the volume is being added up due to the reabsorption of water. So this is known as counter current multiplier mechanism. Current is going against the current, the volume and is being multiplied, and so the pressure is multiplied. So about 99.5% of water is actually absorbed with the help of these only 15% of nephrons, that is juxtamedullary nephrons. And as they reabsorb more and more water, they add up it into the blood. Ultimately, much less water is being left in the urine. Only that much water is being left, which is only required to excrete the waste products out of the body, because solutes, they always need a solvent. So only that much, and concentrated urine is being produced with this mechanism. And as I have mentioned earlier, this movement of salt is in the ascending limb of loop of Henle. It is controlled with the help of aldosterone, or hormone secreted by adrenal cortical hormones. So in this way, Kidney performs its important function of producing concentrated urine, but this does not happen all the time. This is when it is required, like in hot and humid weather, when ADH is released, maximum amount of water is reabsorbed with the help of these juxtamedullary nephrons. It is reabsorbed into the blood, and very small amount of water is excreted into the urine. When there are winters, when enough water supply is there, person is in cold environment, no ADH is required, so no excess water is not being reabsorbed. Juxtamedullary nephron just relax, and excess amount of water is excreted in urine. That's why in winters people they have to go to washroom to for micturition again and again. So this is all about kidney. Another very important function of kidney, despite urine formation, that is excretion and osmoregulation, is that it secretes a hormone, erythropoietin. And erythro means red blood cells, pertaining to red blood cells. So erythropoietin is a hormone which acts on the bone marrow, and it stimulates the process of erythropoiesis, or the formation of red blood cells. That's why people who suffer from renal diseases, who suffer from renal failure, they also suffer from anemia. Their blood cells are not properly being synthesized due to deficiency of erythropoietin. And also a hormone, renin, an enzyme rather, renin is secreted by kidneys, which help to regulate blood pressure. So I think with this you must have understood the structure and function of kidney and the formation of urine. So I think it's enough for today. Stay connected, stay tuned to our platform, OUC Online University courses. And if you have any queries regarding the subject or the teacher, you can contact us at ourchoiceouc@gmail.com. I repeat, ourchoiceouc@gmail.com. Thank you. Take care. Allah Hafiz.